Hello and welcome to the first episode of Odds and Ends. In this video series we'll cover the topics that would be on their own series or on a BitChute exclusive video, but did not make the cut either because they're too short to make a video on their own, or simply put did not fit anywhere. This series will cover 2 to 3 plants or animals that are connected by a common thread. In this case, the first Odds and Ends video will cover 3 South American crops. You'll cover Ialus oleifera, and it's used in crossbreeding with oil palm. You'll talk about the palm tree called Ashanica, found in the Peruvian Amazon, and you'll cover the domestication of various non-quinoa chenopod species found in the Andes. Let's get started. Right now we'll cover, first, the crossbreeding between oil palm from Africa and their close relative, Ialus oleifera, from Brazil. The organization known as CIRAD, currently being run by Dr. Barcelos and Dr. Amblard, did a few crossbreeding experiments, and here are the results. These hybrids were resistant to the disease bud rot. The F1 hybrids produced had an excellent oil to mesocarp ratio, but a low oil extraction rate, below 18% for the best crosses. Further improvements using these hybrids to gradually integrate small amounts of Ialus oleifera into oil palm were also done, in parallel with another program called the BC program in Latin America. The result of this breeding allowed for more breeding for fusarium wilt resistance, leaf miner resistance, and bud rot resistance, with several genes for these traits being found. Another organization run by Dr. Alvadro, Dr. Henry, and Dr. Escobar, in an organization called ASD, likewise did many experiments with hybridization, using both F1 hybrids and back crosses. This led to a commercially viable new breed called the Amazon hybrid that produces 36 tons per hectare of oil, 50 tons per hectare in the best plots of course, and it's partially self-fertile. Now onto the chenopod section. Now onto the Chenopodium archaeobotany, in which we'll cover the archaeobotany of Kanawha and Pico. Dr. Essentrout in 1998 found the earliest known samples of chenopod material within a cave in Peru this cave is called Pantulaca Cave. These samples date back to approximately 3000 BC. This sample could be either quinoa or canafna, but it's unknown which one it is. Canafwa is another species of possibly domesticated chenopodum. It is often found in dump heaps within sites within the Andes. Due to its weedy characteristics, it's speculated to be either a weed or a partially domesticated crop. It also shows no evidence of interbreeding with quinoa due to the fact that it's predominantly self-pollinating in nature. As of right now, this plant's still being grown within the Andes as what's called rustic domesticate, because it maintains several of its wild characteristics such as self-seeding, variable maturity rates, and the ease of shattering. Both quinoa and this species tend to grow together. Pico, on the other hand, has been historically used as a medicine to treat worms. Given that wild or dark seed forms of canafha were also found in that same region 3000 years ago, it is likely that the domestication process began around that time, likely earlier, as both domesticated and non-domesticated forms were still being used. Later research found that the domestication period may have been a little bit later, around 1550 BC, although there's some Chilean samples of chenopod that date 2000 years earlier, although whether or not they're domesticated remains unknown. The Ashinacan palms are a series of palm tree species commonly grown by the Ashinaka people within the Peruvian Amazon. There appear to be various physiological differences between several species that they grow and their wild counterparts. The most domesticated species seems to be Bactris gassipi, which is used for various artifacts involving religious ceremony. It is found in most of these people's myths and legends, and is a staple food source for starchy fruit. and these plants seem to be unable to grow outside human intervention, required them to be planted by various Ashanaka farmers. Although these two species of common palm grown by the Anishinaabe people are cultivated heavily, they were not found in home gardens for these peoples, nor were any other palms, surprisingly enough. Another common species planted by the Anishinaabe people seemed to be Atelia fowlerata. People have been observed carefully selecting different seeds from different parents for planting purposes, further indicating that continuous cultivation and selection are being done by these people on a species. There also seems to be breeding for other properties as well, such as larger leaves for thatch making and stronger wood for different tool use. Although they mostly rely on wild palms for wood, there also seems to be evidence of purposeful crossbreeding between different species to improve their breeds of palm fruit. For instance, there's been found to be a population of Attila phaletra growing in the Saviri village that has traits of both Attila phaletra and Attilia wibberbari, indicating some interspecific hybridization or hybridization between two species. Well, that about covers everything for this episode. Thank you for watching. Hello and welcome to Odds and Ends, Part 2. In this episode, we'll cover the domestication of mink, tobacco, and rainbow trout. Let's start with mink. Mink is a species of weasel called Neovision vision. In Canada, domestication of the mink began in 1866 and expanded from there. In fact, it began in my home province of Ontario. The paper by Dr. Morris of Trent University in Peterborough, Ontario, Canada, hoped to analyze the full genetic diversity of the species, and here are the results. It was found that, despite beginning very early on in terms of breeding for pelt production, there's a wide difference between the amount of genetic diversity between both captive and wild populations, and wild samples found near farms and wild samples in farms have a great level of genetic variation differences. It was also found that there's a low level of variation within functional genes within the American mink. It was found that androgen receptor genes, which regulate the flow of testosterone in terms of behavior patterns, have been altered with only the variants of that gene that correspond to low aggressivity. And various different breeds of mink, from the earliest strains to the latest strains, show a general decline in terms of aggressive behavior. It was also found that the domesticated mink have been bred for faster growth rate for better fur production. As a result, one of the genes responsible for the growth rate, IGF-1, has been mutated through the breeding process, with the mutations being selected for to speed up the growth rate, although it comes with the cost of physical abnormalities such as altered metabolism, altered physical structure, and loss of hearing. While in the wild mink, only a very narrow set of parameters for that gene are allowed. And overall, the captive mink has a very low gene pool in comparison to the wild counterparts. This is due to artificial selection and lots of inbreeding due to the fact that most of the color variations are recessive and need to be bred through inbreeding. As a result, to maintain certain levels of health within the mink populations, the frequent capture of wild mink to bulk up the genome through crossbreeding still occurs to this day. Now onto tobacco domestication. In a paper by Dr. Tushingham of Washington State University, it was found that tobacco was actually domesticated in the South America regions of the Andes between 6,000 to 8,000 years ago. Continuous breeding based on current genetic evidence suggests that the species Nicotiana rustica and Nicotiana tabacum, or common tobacco, are derived from these early domestication efforts. It spread throughout the Americas through the Mesoamerican empires, eventually reaching what would be known as the southwestern United States between 2500 to 3500 years ago BP. although wild tobaccos were used mostly by the Western North American people. Based on current ethnobotanical evidence, tobacco has been historically used primarily for religious or cultural rituals, and very few pieces of tobacco are found in archaeological records due to the fact that they're combusted, ultimately destroying the material. Utilizing different techniques, the researchers attempted to find remnants of nicotine on ancient pipes found in North American dig sites, and they successfully found a few such pipes that had such residues on it. These pipes date back between 2,500 years and 500 years BC. These are found in the mid-Columbia and Snake River regions within North America. These are the earliest known examples of nicotine consumption within the North American region, indicating that the minimum time period of the origin of tobacco smoking within North America is 2,500 BC. Now on to trout domestication. Hello and welcome to Odds and Ends Part 3. In this episode we'll cover Anato, Ofunta, and Horizontal. You'll start with Ofunta. Ofunta, also known as prickly pear, is a genus of different cacti, which includes 188 species, 78 of which are native to Mexico, and at least a few of them are domesticated. Species that are domesticated include Ophantus albicarpa and Ophantus ficus indica, while other species, such as the one seen on this picture. One such key difference between semi-domesticated and wild strains versus domesticated species is that there's a higher pulp amount in the domesticated forms and far more aborted seeds. In fact, the two domesticated species I mentioned before had 50% more aborted seeds than any of the semi-domesticated species, or wild species, of prickly pear. The selective breeding process that the two domesticated species have undergone have also changed their physiology as well. They now rely more so on asexual reproduction than sexual reproduction. There's also found to be a loss in seed hardness in comparison to the wild variants. Ophantia ficus indica has softer seeds that imbibe more water more quickly than any of the wild species, indicating a loss of dormancy often found in domesticated plants, and far fewer seeds of Ophantia ficus indica germinated when planted, further indicating a greater reliance on asexual reproduction, a phenomenon found in many types of tropical fruit trees and asexually propagated crops. As of right now, the two domesticated species of prickly pear include Ophantia albicarpa, which has over 20 cultivars, and Ophantia ficus indica, which has only 15. Now on to the next part. Horizontal is a Chenopodium species that was utilized by the Aztecs and began declining shortly after the Columbian era. It is used to make the two crops known as chia, as in chia pets, chia, and quillette, a spinach-like strain used for vegetable purposes. The genus name for the species would be Chenopodium nataliaceae. It was initially thought that the species was actually a subspecies of quinoa, although later genetic tests disproved such a hypothesis. Based on the genetic tests, this is domesticated purely in Mexico, negating that hypothesis. Based on current genetic evidence, the weed species Chenopodium bellarendii is a likely candidate as the wild ancestor to this species. Since the oldest specimens are found in Mexico, it's likely that, unlike which was domesticated in South America and moved to Mexico, this one was in fact domesticated in Mexico and moved south from there. To further test this hypothesis, Horizontal was crossbred with Chenopodium bellarendii and many other Chenopodium species. The hybrids between Horizontal and quinoa showed limited fertility and rarely set seed, indicating genetic incompatibility. Most of the Horizontal and Chenopodium bellarendii varieties, when they were crossed together, produced sterile offspring, with a few fertile offspring mixed in, surprisingly enough. but several subspecies actually crossbred and produce fertile hybrids with horizontal. Further similarities between both horizontal and specifically the subspecies Chenopodium belarundi, subspecies Shakshi, show great genetic compatibility, and utilizing genetic comparisons, it was found that the species Chenopodium natale, or horizontal, is actually a subspecies of Chenopodium belarundi, which is quite fascinating. Now on to the domestication of anato. Anato is a large tree that is used to produce dye within eastern Amazonia. It is speculated that this species, Bixa aurela, was domesticated from Bixa arborella. There are about 5 species recognized within this genus that are used for this purpose of dyeing: Bixa aurela, Bixa arborea, Bixa excellia, Bixa platycarpa, and Bixa urucana. Still yet another individual suggested that Bixa excellia might be a wild ancestor to anato. Utilizing genetic comparisons, however, they found something quite different. After a complex study of the biology and ecology of the species, it was found that it's more likely that Bixa urucana is the wild ancestor to this species. This is due to the fact that the only trait that separates Bixa urucana from anato is the descendance trait, and the largest population of uncultivated anato are found in the same region where Bixa urucana dwells, in the Paru River. Both species seem to frequently crossbreed with no difficulty, and there are almost no differences between the hybrids and the wild strains in terms of physiology, indicating that anato was domesticated from the Bixa urucana species. In terms of when it was bred and where it's bred, it was found that there are no examples of Bixa species in the Caribbean naturally. As such, it could not have been domesticated in the Caribbean area. As such, it had to be found in continental South America. The first known example of anato in the archaeological finding was dated back to 2400 BP, although a later strain was found in an area called Plum Piece, which dates back to between 3700 and 3600 BP. As such, it is likely that it was domesticated around that time. Well, that about concludes everything. Thank you for watching. Hello and welcome to another episode of Odds and Ends. In this episode, we'll discuss the ethnobotany of mistletoe, ayahuasca, and betel nut. Let's get started with mistletoe. This group of plants are a series of parasitic plants that have the entire root system attached to the xylem and phloem of another species of plant, such as a tree. Although the plant produces its own through photosynthesis, the resources found in soil are instead extracted directly from the plant it's hosted on. Unlike most temperate parasite plants, this species is perennial and lives almost a decade, and in some cases even longer. Since this plant does not seem to kill the host, it is not regarded as a plant pathogen, although it does cause disfigurement in some cases. Mistletoe also plays a huge role in pre-Christian and post-Christian folklore and mythology, and was well studied by various philosophers in ancient Greece. Pliny accurately described the life cycle of the species, where the fruit of the mistletoe is eaten by birds who then poop it out on another species of tree, allowing the seed to sprout and grow into the tree itself. The ancient Celts believed the mistletoe was of divine origin, and many Christmas traditions that involve mistletoe are derived from these ancient traditions. Many cultures also used them for medical purposes in ancient times, but they also contain different poisons. As such, those past cultures who used mistletoe also had cures for mistletoe poisoning as well. It was used historically to treat heart disease, syphilis, epilepsy, ulcers and sores, and the Black Plague. One unique aspect of mistletoe is that due to the fact that it's attached to a host species, the plant accumulates compounds produced by their host organisms, such as mistletoe is grown on coffee trees, having caffeine within their bodies. And various sources say that it is hallucinogenic, although other hosts say it's not. This may be due to the fact that it takes up chemical compounds produced by their host plants, thus allowing them to take on the traits of their hosts. Now on to the next plant. Ayahuasca is a beverage obtained from extracting substances from the plant species Banastropis capi or Psychorta viridis. Ayahuasca is derived from the words aya meaning spirit and huasca meaning vine. It is commonly used in religious cults in Peru, Bolivia, Brazil, and parts of Ecuador. Consumption of this product seems to cause effects similar to LSD or similar drugs. Prior to Colombia showing up, the indigenous cultures of that society utilized shamans, people who had a connection to the supernatural world, supposedly anyways, and used such beverages to go into deep trances where they would interact with spiritual entities, or more likely, hallucinations brought on by the intoxicating beverage. Utilizing the visions obtained from this plant decoction, the shamans would come up with cures for different diseases and ailments, and create different forms of art. This became the origin of a new concept called the Professor Plant. In this conceptual idea, plants directly inject knowledge into people by consumption. I don't really believe in that sort of thing, but there is that. Various groups in South America still use ayahuasca for different religious purposes. These new cults are a mixture of Catholicism and native folklore, as well as African Brazilian folklore. The most well-known cult within Brazil that involves ayahuasca is the cult known as Abrucá, a cult created in 1945 by Daniel Pereira Matos, also known as Maestre Daniel. He initially was part of another cult called Santo Daime, where he lived for a decade before splitting off and founding his own cult in the city of Rio Branco. Now on to the next plant. Now on to the use of betel. This is a concoction of acrea and betel pepper and lime. These three plants, when combined together, create a narcotic effect. The first plant used is called acrea, a genus of palms which include the domesticated betel palm. The species is easily grown and often becomes weedy, and the fruit are easily picked as well. The next plant is called betel pepper, which is a member of the pepper family, as in black pepper. In fact, it's a sister plant to black pepper, known as Piper beetle, a sister plant to Piper nigrum. These plants are typically grown from cuttings by the people within that region. Finally, they make lime from limestone by burning shells. Due to the narcotic properties of this mixture of plants and limestone, this mixture of plants and lime is often used for divination purposes or for treating certain illnesses. It is also used in folkloric religions as offerings to the divine and for various ritual purposes. And it's also consumed socially, often around a campfire amongst different groups of people, where the chewed material is exchanged between people, as well as ingredients, indicating a heavy communal use for this plant, possibly to use the hide to create greater bonds between kinsmen and tribal peoples. Well, that about covers everything. Thank you for watching. Hello and welcome to another episode of Odds and Ends, Part 5. In this episode, we'll cover several crop plants, specifically jojoba, service tree, Kadako plum, and Maori potatoes. We'll start off with the plant species jojoba. The species is called Simsosia chinescens, or by the common name jojoba. It's a deciduous woody perennial plant native to southwestern North America. This species produces an oil that has the properties of a liquid wax, making it suitable for lubricating machinery at temperatures up to 300 Celsius. However, the wild populations provide inconsistent qualities and quantities of wax. As such, in 1925, the first attempt at domesticating this crop happened in the Boyce-Thompson Southwestern Arboretum in Arizona. Independently, other countries also began tinkering with the plant to try to domesticate it, such as Argentina, Australia, Chile, Egypt, Israel, Mexico, and Peru. In Argentina, little domestication has happened, but they've cultivated over 3,000 hectares of jojoba for oil production. In Australia, by the year 1978, selectively bred cultivars suited to Australian conditions were developed, formed by the union of an Australian government research agency called the CSIRO and the New South Wales Department of Agriculture in the late 1970s and 1980s. In Brazil, breeding experiments between 1977 and 1980 also developed further varieties of jojoba that could be grown without irrigation. And to this day, Brazil is still a major player in jojoba breeding, with many commercial cultivars coming out of that breeding program. Ironically, due to the similarity of this plant's wax to sperm whale oil, the cultivation of this crop reduced the need for whaling by a considerable margin. Now on to the next crop. The next species on our hit list is one with a lot of mystery, Sorbus domestica, or the service tree. This species is unique in that, although it's in the genus Sorbus, it cannot form hybrids with even its closest relatives. It has been historically used in both fresh consumption processing, honey production, and cider production, as well as folk medicine. This species occurs in Central Europe, including Slovakia, Austria, Chechnya, and the White Carpathians, which acts as its most northern edge for this species. Its edible fruit comes in a wide array of different shapes, as seen on this picture, and it varies between individual trees. Based on a study of a large population of service trees in the Carpathian area, seems to suggest that many of these populations are managed landscapes / ancient orchards of some sort. Since in this region, very few forest biotypes are found, and most of the specimens are of heavily cultivated forms. Also in that area, it's been known since the 15th century that gardens and orchards were used to grow and produce these plants for their fruit and for their wine clarification properties. There is also a high proportion of single individuals within certain parts of this region, indicating that the current range in the Carpathian area of the species represents a long-forgotten village and orchard location, where the people left but the trees remain. As of right now, it's unknown from where and when this plant was domesticated, and is still to this day being debated. Much of the history has been hidden as well due to the fact that much of this location has been ravaged by socialistic policies of the 20th century due to field consolidation. Since the species is highly fragmented throughout Central Europe, it is concluded by a researcher that all specimens, even those of non-natural origin, should be preserved for future breeding purposes and conservation purposes. Now onto the Kakadu plum. The scientific name for a species is Terminalia ferdinandia. This is a species of fruiting plant found in northern and western Australia that has fruits used for different food and cosmetic products. In combination with the high vitamin C content ranging from 0.2 to 5.9%, and the ease of propagating the species using both seeds and grafting, and the discovery that adding irrigation actually increases the rate of which the fruit grow and mature, led to interest in breeding this crop. And so in 2018, August 10th, the long since established Kakadu plum industry formed a breeding project with an organization called Farming Together, as well as a company called Kindred Spirits Enterprise, and the government organization CRCNA, to breed improved cultivars of Kakadu plum for the many needs and interests surrounding this unique fruit. Now on to the next crop. Now on to the last part of our video, Maori potatoes. For the longest time it was unknown how the Maori got potatoes, since potatoes are native to the Americas and Australia is nowhere close to that location. In a paper by Graham Harris in 2005, And it's also unknown how they grow them there because potatoes, although they live in the tropics, they live in the very high altitude areas that are very temperate, and New Zealand is fully tropical. Here are the results of his study, however. In terms of textual evidence, many of the journals specifically mentioned that they were growing potatoes in New Zealand when they got to New Zealand from the Old World, and had already made mythology of how they got it, including the mythical Tiriki Tarango, who obtained potato from a hidden world. There's also listed 15 varieties of potato that appeared, according to the natives, prior to the appearance of settlers. Another bit of mythology called the Song of Waha, I'm definitely butchering that name, also mentions mythology surrounding potato. On the linguistic side, the Maori name for the potato is called puru puru. Puru is the direct origin of potatoes, but not from the native language of the Peruvian people, who called it Quichua, but it's a derivation of the local chief Biru, named by the Spanish conquistadors. Now on to the cultivars de jour. The cultivar renca has elongated tubers that are small and purple. It's one of the oldest known Maori potatoes described by Harris, and has many wild character traits similar to the European cultivar Congo, which likewise is known to have wild traits. Both have heavy similarity to the native Andean potatoes, such as the Chian Kuchan, making it possible that it was either brought over from South American ships via the Spanish or direct trade between Peru and Australia. In other studies, cited by Graham Harris, it was found that there are no examples of fossilized or preserved remnants of potato pollen or tubers, indicating that potatoes, in this context, are not of pre-European origin. And based on the fact that they sow seeds regularly from potatoes to breed new varieties, the Maori potatoes are likely derived from stock brought over from the Spanish conquistadors to New Zealand, which were then allowed to crossbreed to produce many new varieties in the process of redomestication, with the mythology being younger than expected, and the stories from the travelers being just that, stories. But nonetheless, altering a species that's warm-temperate to a fully tropical environment is an impressive feat of the Maori people. Well, that about covers everything. Thank you for watching. Hello and welcome to Odds and Ends number 6. In this episode we'll cover one of the most prolific rose breeders in modern days, Ralph Moore, the master of miniature roses. Ralph Moore was born in 1907, January 14th, in California, to a family of backyard rose gardener. His first plant breeding project started in the 1920s when he bred new types of myrtle and lilac. In 1935, he saw his first miniature rose called Roletti, a unique Swiss breed of rose. Using $800 in a dream and no education, he formed Sequoia Nursery in 1937 and learned from scratch the art of rose breeding. And I will cover his long and storied history throughout the breeding process, specifically his miniature roses, which gained him great popularity and acclaim. Although it's believed that most of the miniature rose population at that time was developed from the Chinese miniature rose, Rosa Roletti, Mr. Moore bred his own varieties from scratch using different breeds such as Oakington Ruby, Tom Thumb, Cecil Brunner, and various species. Although his primary breeding stock was the cultivar Gwynnie, a climbing hybrid tea with a red flower, the species Rosa Wichieria, and the species Rosa multiflora, and of course Oakington Ruby, a chance seedling of Rosa Roletti that was found by a gardener named Mr. Bloom. 15 of Mr. Moore's early breeding program cultivars were derived from Oakington Ruby. These include Centennial Miss, Candy Pink, Diane, Red Germain, Westmont, and many others. Most of these varieties were made by crossbreeding Oakington Ruby with a crossbreed between Rosa Wichieria and Rosa Floridora, or forced self-pollination between Oakington Ruby and itself. He further improved his own cultivars by crossbreeding them with an old hybrid perpetual called Ferdinand Picard, a striped rose. One of the most recent breeds was the cultivar Just For You, which had all the beauty of a classic hybrid tea rose in a small compact form. Mr. Moore had also taken a big risk, since he started out when the first miniatures were just appearing on the market and hybrid teas were still king. But thanks to his foresight, his miniatures managed to sell 4-to-1 in comparison to hybrid teas. In 1996, he also bred one of the first dark red varieties of miniature rose, called Cherry High. Mr. Moore also created the first hybrid rugosa roses in California, such as Topaz Jewel, Playgirl, and Red Cascade. And in the 1990s, he also released his Halo series, a series of small, single-petal miniature roses. In 1965, he likewise bred various types of roses such as Green Ice, which are roses that have green flowers that fade into white as they open. Magic Carousel, a hybrid tea variety with a blend of various reds and reddish colors. Mary Marshall, a miniature orange hybrid tea, and Rainbow End, a miniature rose that blends yellow and red together. And the Clammy Mini, Rise and Shine, a yellow-colored rose as well. But his oldest cultivars occurred in 1954, where he bred Renee, a thornless rose that repeat blooms. This was derived from Cecil Brunner. It wasn't until 1957 that he abandoned everything to focus on miniature roses. In 1975, the first refined form of miniature rose came out of his breeding program. This was referred to as Dresden Doll. It was one of the first miniature moss roses generated. Prior to that, he tinkered with other moss roses such as Golden Moss, Pinocchio, and various other cultivars. One such seedling called 125910 was crossbred with the cultivar New Penny to produce fairy moss, the first known miniature moss, which was then used to breed Dresden Doll. He also tinkered with the Floribunda style roses by crossbreeding miniature Floribundas with the centifolia rose Chapelle de Napoleon to breed the cultivar Crescent Sweetheart in 1988. In 1999, the Vineyard Song rose was introduced, one of the first purple miniature roses. This was a crossbreed between the miniature rose Little Chief and the old multifolia rambler called Violet. Mr. Moore likewise continued to work on various other cultivars, leading to the creation of the miniature called Lemon Delight. This is because the moss on the buds of this flower were scented with lemon. It was a crossbreed between fairy moss and gold moss, so it was also lemon colored as well to add to the intrigue. In 1988, likewise, the first miniature scarlet red rose with moss on the buds, scarlet moss was bred. The mossing on the sepal smelled intensely of blossom, although the flowers themselves did not have any fragrance, but did have a bright red scarlet flower. He used the climbing rose Dortmund to breed scarlet moss. The other amateur plant breeder, Paul Barton, also took frequent trips to this nursery where he found various other seedlings, including a strange seedling that starts out pink but become green over time. In 1998, the rose cultivar Jock Williams was bred. It's a crossbreed between the hybrid between Yellow Jew and Tamango and Strawberry Ice, one of the very few bicolored miniature roses. Ralph Moore died on September 14, 2009 at the age of 102, ending his long and illustrious career as a prolific rose breeder. Well, that book covers everything. Thank you for watching. Hello and welcome to Odds and Ends number 7. In this episode we'll cover the domestication of Yacon, Oka, Aronia, and Haskap. You'll start with Yacon. Yacon is an ancient Andean crop whose scientific name is Smolalanthus sonifolius. Yacon is a polyploid species of a possible hybrid origin. Based on the current biology of this domesticated crop, this plant species has infertile seeds and is reliant on asexual propagation by people to propagate this crop. The yacon produces large edible tuberous roots, traditionally consumed as fruits similar to sweet potatoes in appearance, with a juicy and a rare sweet taste, but lacking in starch. The common yacon exists in proximity with 4 other species closely related to this species, which includes Psammophis cybercus, Psammophis macrocyphus, and Psammophis cononatus, and Psammophis sulfurfructus. It is likely that the common yacon is derived from at least 2 of these species. In a study using the wild species of yacon and the cultivated yacon itself, it was discovered that although it's considered a semi-domesticated plant, all the wild species have significantly more pith and fiber in comparison to the domesticated form. The cultivated form can also withstand several days of transport and hold high levels of water and sugar, indicating further domestication than first realized. The species S. cybecus seems to have the most reducing sugar amongst the wild species, indicating that it's one of the parent species, although it's not the sole species since it also has a larger, if not the largest, xylem conductive area of the 4 species, making it very fibrous and unpalatable for most people. Now on to the next crop. Oca. The species Oca tuberosa is another polyploid North American crop. This species is octoploid, but most of the regular wild species are diploid, with only a few wild polyploid forms, and the majority of these close relatives do not produce tubers, indicating that only 4 species were used in the breeding of this domesticated species. These include the identified species Oxalis pinchensis and Oxalis chinsagensis, and 2 unidentified species labeled Lima WT and Bull WT. The former is found in the Andes in the Lima department, the latter being found in Bolivia. Although Oxalis tuberosa is currently considered one single species, unpublished data from E. Eschenweiler seems to indicate 2 distinct genetic groups, and traditional growers of Oca have 2 different names for it. The Wayek cultivars are sweeter in taste and were usually cooked fresh after a few days to sweeten in the sun. The Akaya type is primarily used for processing purposes to make dried tubers that can be stored for long periods of time, and are sour in flavor. Genetic analysis using genetic markers indicate 2 separate domestication events, possibly from different species. And here are the results of this study by Dr. Eve Emschweiler. Based on current genetic evidence, the Bull WT species and the Lima WT species are in fact distinct species, genetically distinct from both each other and the other 2 named species, Oxalis pinchensis and Oxalis chicensis. The genetic analysis also suggests that the Wayek and Kea type cultivars show distinct domestication histories and possibly different origins for their polyploidy. The speakers of both different languages, Quichua and Aymara, in the South Americas, likewise distinguish sweet oca from sour oca. It is concluded that both these strains may have different origins, and in fact, may have been an example of independent domestication and independent polyploidization, although further genetic sampling may be required. Since this species is octoploid, and there's no examples of tuber-bearing wild species with octoploid genomes, it is likely that this is an allopolyploid, the hybridization between multiple species. It was early suggested that Oxalis pinchensis is one of the genome donors to the octoploid Oca, but current results suggest that O. pinchensis is the least similar to Oca. It is plausible that integrative hybridization may have played a role in the development of the species, but as of right now, Oca pinchensis is not one of the species. The species Bull W/T and Oxalis chilgensis are relatively genetically close to each other, as well as Oxalis tuberosa, indicating that these two may have been parents in the crossbreeding process. And Oxalis chilgastensis had the most genetic traits shared with the Wyak Oka samples. And the bull WT strains are also genetically very similar to Oka, further indicating that these two were probably parents in the production of these cultivars of Oka. So as of right now, 2 of the 4 candidates for genome donors to the octoploid species have been found, but the other 2 candidate species for Oka remain a mystery. Now on to the next crop. Now on to Aronia. Aronia melancarpa, or black chokecherry, or just plain Aronia, is a species native to North America, along the east coast of Canada and United States. It was cultivated mostly as an ornamental, but it's popular in medicinal and culinary uses as well, especially in European countries. The first wild species of Aronia were introduced into Europe at the end of the 18th century. And the first breeding program was developed in Scandinavia and Russia. Farm cultivation on industrial scale began in the Altai Mountains, Moscow, and Petersburg. This species is commonly grown as a means of making juices as well as producing health food substances. At present, Aronia is commonly grown in Central, Eastern, and Southern Europe as a backyard crop, and commercial plantains are based on the cultivars Nero and Galganta. These are high-yielding cultivars with great resistance to harsh conditions found in Russia and other parts of the world. Due to its beautiful leaf color in fall, it's used extensively as an ornamental plant, with the cultivars Viking, Aron, and Huggen being the most popular forms of this species. In the United States, the cultivars Autumn Magic, Mackenzie, and Morton are the most popular forms of this ornamental plant. Now on to Hascaps. Hascaps belong to the species Lauronica caerulea, a species of honeysuckle, native to the northern boreal forests of Asia, Europe, and North America. It is primarily found in low-lying wetlands or high and mountainous areas. The first domestication efforts happened in Russia, starting in 1913 to 1915. The project was then shelved until the 1950s in the Vavilov Institute and the Siberian Horticultural Institute. The first 3 cultivars of Hascap were developed in the 1980s, bred by the late Dr. Maria Emplakova, former head of the Fruit Crops Department in the Vavilov Research Institute. Over a 25-year period, several cultivars were bred, resulting in the creation of 68 cultivars. At least 60 of these have been commercially generated and propagated. These cultivars were derived from populations of wild Hascap found in the northern parts of Russia, as well as the European parts of Siberia and the Far East. The tetraploid forms of this species were found to be extremely useful for domestication purposes. These were found in the Kuril Islands, as well as parts of China and Kazakhstan. The breeding program was made to increase berry size, flavonoid levels, and depth of dormancy. One cultivar, Savast, was a maternal parent for many named cultivars, such as Sinyaya, Pizza, or Bluebird, Gobaya Vereteno, translation light blue or sky blue, Flaka, translation violet, and the cultivar Marina. The cultivars were selected from different regions for different purposes. The strains found in the Kamchatka Peninsula had high levels of winter hardiness, large fruits, sweet sour taste, and high absorbic acid. The Hascaps of the Kuril Islands had deep dormancy of buds, high winter hardiness, large fruits, and late maturing capabilities. The maritime areas within Russia had strains that were early ripening, high yielding, and high in bioactive compounds. The Altai and Saiyan mountains had high yield, fruit that wouldn't break, and high levels of drought resistance. And in Northern Europe, there was a deep dormancy and high winter hardness and non-chattering traits as well. In Japan, domestication of Hascap began in the 1960s and 1970s. by the Hokkaido Prefectural Agricultural Experiment Station and a farmers' co-op in the Chichos region. It was popular in Japan early on, but due to the high cost of labor, it declined, becoming a smaller industry than expected. In Oregon, Dr. Maxine Thompson and Mr. Jim Gilbert began tinkering with the specimens from Japan and Russia to breed new varieties. The University of Saskatchewan obtained many of these parental cultivars used by this breeding program, obtaining 35 Russian cultivars, 70 plus Japanese cultivars, and hundreds of seedlings from Dr. Thompson's breeding program in Oregon, as well as 6 wild Coral Island types and 600 specimens of wild hascap found in the boreal forest of Canada, obtained approximately 8,000 seedlings from controlled crosses in 2008. In 2007, Canada released its first cultivars, Borealis and Tundra, bred from Russian cultivars. Well, that about covers everything. Thank you for watching. Hello and welcome to Odds and Ends, Part 8. In this episode we'll cover the history of seaweed propagation. The common seaweeds being produced are red seaweed, brown seaweed, and some green seaweed, but the red and brown are the most common. The red seaweeds are part of the genus Piporia, and the brown seaweeds are in the genus Saccharina and Uderaria. The primary mode of reproduction for the green and red seaweeds are asexual reproduction via meiospores, making them easy to mass propagate. Kelps or green seaweed, on the other hand, do not regenerate from pieces or from meiospores. They have a sexual part to their life cycle. The asexually propagated species are cultivated by vegetative propagation based on chunks of the seaweed itself, which is divided up and propagated asexually. Now on to the history of this process and the optimization of mass-producing plants. It was discovered in the brown seaweed species Saccharina latisma, revealed that by changing the temperature or by chopping up a part of the seaweed called the thallus, can cause the seaweed to release massive amounts of meiospores, which can be used to generate a legion of asexual spawn. It was also found that the use of red light can prevent sexual propagation from happening in brown seaweed as well, and the production of sexual embryos through fertilization is triggered by white light. During the early periods of propagation, the spores of the seaweed are attached to ropes which are kept wet in seaweed nurseries. Once the young seaweeds achieve a size of 1 to 2 millimeters, they are then moved to open water. This process can take up to 8 months. It was later discovered, however, that certain textile sheets can double the growth rate of all 4 types of spores, including meiospores, gametophytes, and sporophytes. It was also discovered how to asexually propagate sporophytes, which grow even faster than meiospores and gametophytes. In such genera such as Galcerina, which produce almost fully grown propagules, and the genus Campophyllus and Euchima, which can be propagated on tissue culture systems, even faster biomass production has been created. Later on, a new method called the tubular net method, modeled after the mollusk farms, was used to replace ropes, improving the amount of seaweed propagules you can add onto a piece of rope mass. And it's better for the environment since the seaweed does not invade the current environment. It's also very efficient in fast-growing species because side branches will quickly grow out of the mesh in a very quick fashion. And this can happen before sediment overgrowth or epiphyte overgrowth, depending on the area, can happen. However, the seaweeds are still subject to natural disasters such as typhoons and storms, which can damage or destroy entire crops. The only solution to that problem would be growing them in tanks, which we'll cover right now. Growing seaweed on land is a very difficult process, since the cost of keeping sealings in tanks is very expensive and lays with difficulties such as acclimation pressures and possible disease control. The first example of on-land managed seaweed cultivation is Irish moss, or Chandra's cripus aquaculture in Charlesville, Nova Scotia, Canada. The various problems that need to be overcome included improving photosynthetic rate, reducing zinc contamination, disease control, and cultivar selection. To that end, that company that produced Irish moss within a tank utilized drone monitoring, real-time adjustments of air delivery systems, compressed air agitation in each tank, continuous alteration of pH using carbon dioxide, with carbon recovery up to 90% in large-scale cultivation tanks. Issues with water pipelines were solved by accessing deeper and cooler seawater, which contains low levels of sediment. Various measures were developed to improve mechanical sorting and handling techniques to improve the efficiency of seaweed production in such a situation, with most of the seaweed being imported to Japan. To this end, extensive work in seaweed breeding, biology, mechanical engineering, and marketing were done to make this plan a possibility. In more recent years, a new system was developed called the Integrated Multi-Trophic Aquaculture System, or IMTA. Through the use of this system, seaweed-based farms on a full scale have achieved commercial success in areas such as China, South Africa, and Canada, and more recently, Israel. As of right now, very few species have been optimized to the system, with the species of the Galcerina genus and Ulva genus being two such examples, with these species having been fully analyzed for optimal growth, optimal light intensity, temperature, water motion, plant density, nutrient enrichment, and genetics. Such technologies have also been used to develop systems for other aquaculture projects. For instance, one such Grasserina species was bred and developed for the function of acting as a biofilter for fish poop in aquaculture systems. As such, the integration of the IMTA system into other fish farms has also improved the production of fish in land-based operation. With the species Ulva lactua, they've reduced the amount of nitrogen within sea urchin tanks and fish ponds. As such, in land-based fish pond and sea urchin farms, the cultured seaweed assimilated 74% of the nitrogen from said fish ponds and sea urchins, and the seaweed could be used as a food source for the sea urchins, further improving the efficiency of land-based systems for fish farming. A similar phenomenon was also found in fish. Now on to the history of disease control within seaweed culture. One of the biggest problems in production of kelp and other seaweed species are the infections by various oomycetes such as Pythium and Olaplopsis, as well as red rot and other diseases, which spread quickly in large-scale populations. In smaller operations, people can do regular acid washing techniques to prevent or reduce the growth of these pathogens. Similarly, other species such as epiphytes, herbivores, and other strange substances can cause losses for seaweed production facilities. Drying and pH control can be used in Phytophthora, or the red seaweed, as well as increasing the density of seaweed to minimize the presence of epiphytes. Later discoveries, such as the extracts of different brown seaweeds, can trigger immune responses within other seaweeds that alter the growth of different seaweed species, making them more resilient against different problems such as epiphyte and abiotic stresses. These biostimulants seem to act as a trigger for the immune response and as a growth promoter within various species of seaweed. As of right now, this has only been seen in the Capaceus genus of seaweed. The mechanism is an interaction between the beta-1,3-glucan found in such extracts and the beta-1,3-glucanase enzyme present in the Capophycus species. Well, that about covers everything. Thank you for watching. Hello and welcome to Odds and Ends number 9. In this episode we'll cover mung bean, vetch, and grass pea domestication. We will start with mung bean. The wild form of mung bean came from Australia. It's called Vigna radiata sublata. This species is found, like I said before, in Australia, specifically the tropical and subtropical regions of northern and eastern Australia. At some point it was moved to India and domesticated 3,500 years ago, based on the highest level of genetic diversity being in India. The land races of cultivated mung bean found in Africa and other Asian countries other than India seem to be derived from the original Indian population. A closely related species called black gram, which was also domesticated in India from the wild ancestor Vigna mungo variety sylvestris. This cultivated species has been crossbred with mung bean in order to improve its genome and add new traits. Now on to the next crop. Next we'll cover common vetch. Known by the scientific name Vecchia sativa, it is a highly variable species with many populations that in the wild overlap, allowing for interbreeding between populations, although this happens rarely since this is a primary self-pollinating crop. The domesticated form of common vetch, known as subspecies sativa, is obscure, but it's speculated that the subspecies nigra is a primordial wild form that the common vetch complex was bred from. This species has the widest range of distribution and genetic diversity within the Mediterranean basin. In archaeological context, there is inconclusive evidence of the domestication of common vetch due to the fact that it's impossible as of right now to differentiate definitively wild, weedy, or cultivated forms of vetch, as seed size overlaps in all three forms. But the earliest known samples of vetch were found in the Tel Abu Harain site in Syria. These specimens date between 9,200 years ago BC and 8,500 years ago BC. And another set was found in the Ersomatic Neolithic site, which dates between 7,500 BC and 6,500 BC. Further evidence was found in the Kant Hassan III site in Turkey, which is dated around 6,500 BC. In that same location, bitter vetch and lentil were also found. In one of those digging sites, 8 seeds of einkorn were found alongside 40 seeds of vetch and 3 seeds of lentil. Most of the plants surrounding the vetch were wild, indicating that this was likely wild-collected specimens. In Neolithic sites in Bulgaria, Hungary, and Slovakia, vetch was found amongst lentils, which date between 4,728 BC and 4,770 BC. In another dig site within the Eastern Europe subgroup, a combination of bitter vetch and common vetch were found that were dated around 2900 BC. While it is plausible that domestication of vetch may have occurred in Europe, it is still possible that these were weedy or wild forms, since there is also circumstantial evidence of early movement of bitter vetch, common vetch, and lentils together. Based on current textual evidence, it is likely that vetch was domesticated in Rome, and from there the domesticated form moved to Europe. Based on the insufficient genetic or archaeological evidence, this will have to do for an explanation. Now on to our last crop, Laphris sativus, or grass pea. This plant has not been cultivated for quite some time, and its origin is very obscure. The most we know is that it's somewhere in Southwest and Central Asia. This species' wild ancestor remains unknown, but the best candidates are Laphris cicerea or Laphris myrmeticus, as well as Laphris bellocarpus and Laphris pseudodicaria, due to the close morphological similarities between Laphris sativus and these 4 other species. As of right now, the current distribution of grass pea is identical to that of lentil, where it covers the Mediterranean area, Ethiopia, and the Indian subcontinent. The earliest known specimens were found in the Neolithic era materials in the Balkans, as well as the Near East in Bulgaria. And once again, there's no distinguishing between the wild and domesticated forms due to overlapping seed sizes and similarities between both groups. This indicates that there was some crossbreeding between wild and domesticated forms at the time. The first known example of some form of cultivated Laphorus was found in a pile of bittervetch seeds dated around 7200 BC, indicating that it was possibly a weed. Cultivated grass pea, lentil, and pea were reported as rare at Iraq, but were still found there. These specimens were dated around 6750 BC. In Turkey, specimens have been found between 5800 BC and 5400 BC. In Iran, specimens have been found between 5500 BC and 5000 BC. And in cultivated lentil sites, a small number of grass pea seeds are found amongst pulses that date between 4928 BC and 4770 BC. In Greece, however, equal numbers of grass pea and lentil were found in that site that date between 3500 BC and 4000 BC. This suggests that cultivation was underway in Greece at that time period, and was likely domesticated there. In the site Tel Bazamon in Iraq, grass pea was the most common pulse found in that location. This location was dated between 2100 BC and 1800 BC, further indicating domestication at that time period. It likely faded away sometime during the Greek era, since the Romans never mentioned grass pea, indicating some form of domestication in the mid-Bronze Age before being outmoded by other crops in later years. Well, that book covers everything. Thank you for watching. Hello and welcome to Odds and Ends number 10. In this episode, we'll cover the possible future of nitrogen-fixing corn. To that end, you'll be studying a paper by Dr. Alan Van Denzi, who is tinkering around with the possibility of breeding nitrogen-fixing corn. Many different grass species, such as Miscanthus X giganticus, some millets, and some sugarcane specimens, there are associations between their roots and microbes called diasphoric species. They're similar to nitrogen-fixing bacteria in legumes, but in different families. In previous studies by Dr. Triplett, Dr. Estrada, and several others, it was suggested that ancient primitive maize or corn landraces may have unique interactions between the disotrophic bacteria. In a landrace called the Sierra Mix landrace, found in Oaxaca, Mexico, a species of nitrogen-fixing bacteria called Bulkhuria was found. It was found that the Sierra Mix landrace had on its brace roots a mucus substance that coated this root system that allowed for various bacteria to grow on it. It was analyzed by the researchers and found various species of Rhizobium, Burkholdia, and Pseudomonas bacteria on it. Brace roots, by the way, are roots you sometimes see in corn that are used for anchoring the corn plant to the ground. The Sierra Mix landrace was further studied, and it was found that, unlike other corn plants where brace roots only grow for a certain period of time before stopping, the landrace Sierra Mix roots continue to grow, increasing the number of brace roots 3 to 4-fold. These roots secrete a mucus that is rich in the chemicals arabinose, fructose, and galactose, free sugar derivatives that are readily available to bacteria. On the screen, you should be seeing an image of both the brace roots covered in mucus and the brace roots on a normal corn plant. It was found that, regardless of where it's planted, the Sierra Mix maize can fix nitrogen from the air directly. It is unknown how it does this, but the two working hypotheses are the seeds contain the bacteria within the seed's body, allowing for the bacteria to grow alongside the corn crop, generation after generation. The second hypothesis is that, through the mucus, Sierra Mix can recruit different bacteria species to substitute for the ones they lost in their native region. The mucus was further analyzed and it was found that 3 species were the primary source of nitrogen fixation: H. cerocobidae, A. brasiliensis, and B. umami. Through other studies, it was found that, depending on the environment, the microbes found within the corn mucus can actually fix 30 to 80% of its nitrogen. The basis of this trait seems to be either environmental or seed-borne, in terms of how it manifests, but the ability to produce the mucus seems to be a genetic trait. In the wild ancestor of corn, teosinte, a similar mucus is found, which contains a similar number of nitrogen-fixing species within its aerial root system. This may indicate that there was crossbreeding between the wild ancestor and post-domesticated corn in that region, allowing for some of the wild traits to be incorporated into the common corn germplasm within that region. This could plausibly mean that improvements in nitrogen efficiency could be accomplished in corn, with a novel mechanism to replace much of the synthetic fertilizer we use with naturally fixed nitrogen. However, further breeding programs would have to be done in order to improve it for a commercial facility. For instance, these land races are 16 feet tall, making them ill-suited for combining in a modern corn system. Likewise, they will likely have to sequence the genome of these cultivars to search for the gene or genes that allow for nitrogen fixation. And here's me speculating, there could be the possibility that this trait is induced by specific bacteria that's bound to the seeds of this land race, in which case it wouldn't be inherited but be an epigenetic phenomenon induced by the bacteria itself, interacting with the genome of the corn plant. But again, that's just me speculating. Regardless, there's probably a long way to go in terms of getting a nitrogen-fixing corn plant to the market. Well, that about covers everything. Thank you for watching. Hello and welcome to Odds and Ends number 11. In this episode we'll cover biofuel, production, history. We'll cover the history of bioethanol followed by biodiesel. Biogas has been covered in a BitChute exclusive, link in the description. And algae-based fuels will be covered in another BitChute exclusive video. Let's start with the history of bioethanol, also known as ethanol. All alcohols are chemicals of an OH group, such as methanol, propanol, butanol, and ethanol. The first use of ethanol is speculated to be by the Chinese in 9000 BC, as pieces of pottery have residues of ethanol on it. For the majority of human history, ethanol was used by another name, alcoholic beverages, or booze, and was used for its usual purpose. Ethanol's use as a fuel in the United States was realized, however, in 1896 when Henry Ford built the first automobile to run on 100% ethanol. In 1906, a $2 per gallon tax on ethanol created during the Civil War in the US was removed, allowing for larger scale use of this automobile. This use of ethanol as a fuel source ended with Prohibition in 1919. 14 years later, interest in ethanol as a liquid fuel additive became important again, with the creation of gasoline, creating a successful hybrid gasoline. It was however cut short due to World War II, when the excess grain was needed to support war victims overseas. This idea was once again revitalized in 1973, because lead, although a great fuel additive to improve fuel efficiency, was, well, lead. Lead was replaced with methyl tertiary butyl ether, or MTBE. MTBE was later found to be toxic as well, so they replaced MTBE with ethanol. In the late 1970s and 1980s, due to gasoline shortages, these hybrid fuels were exempted from federal fuels taxes. This ended when petroleum prices once again dropped. Interest increased again in 1990 with the passage of the Clean Air Act as a means of reducing carbon monoxide emissions. In 2007, the US federal government required a new law that require an increased use of renewable fuels, which includes a stipulation that bioethanol need to be blended with 100% of the gasoline used by 2022. Due to the fact that most cars are geared towards fossil fuel use, not ethanol use, most combustion engines in the car can only have fuel that's supplemented with 10 to 15% ethanol, without altering construction or operation of the engine. As of right now, most of the ethanol is produced primarily from corn in the United States. In most of the world, however, bioethanol comes from two sources, corn or sugarcane. Prior to corn becoming the mainstay, sugarcane was a primary source of ethanol for biofuels, with most of that sugarcane production coming from Brazil. Even today, Brazil has developed a system where 40% of the fuel they use is of sugarcane-derived ethanol origin. But as corn improved, it was found that you could produce more ethanol from corn than from sugarcane, and so the US took over the biofuel market for bioethanol. Now onto biodiesel. Biodiesel starts off, ironically, with the first diesel engine, created by Rudolf Diesel in 1896. The original diesel engine had an efficiency of nearly 75%, which was much higher than previous steam engines in use at that time period. And it ran off an ingredient near and dear to many different types of chefs and cooks: peanut oil. Despite the use of peanut oil as a biofuel, it was eventually overshadowed by regular diesel made from petrol. This was due to the declining prices of petroleum-based fuels in the early 1900s. But of course, research never stopped into biofuels, or biodiesel, completely. People became interested in biodiesel again in 1973 with the oil crisis, caused by embargo placed on the US by OPEC. Unfortunately, the early generations of biodiesel were too thick or viscous to be used as conventional fuels for diesel engines. However, in the early 80s, researchers in South Africa unlocked the clue to lowering viscosity in native biodiesels. These researchers developed a chemical transformation technique that broke down the triglycerides found in sunflower oil into simple hydrocarbons called methyl esters. Methyl esters had lower levels of thickness or viscosity than regular biodiesel, and as such works well in diesel engines. From this process, a chemical technique called trans-esterification was created, which allowed any plant oils or animal fats to be broken down into methyl esters. Trans-esterification is a reaction of fats and oils with alcohol and a catalyst to create fatty acid methyl esters. When triglycerides react with alcohol in the presence of a catalyst, fatty acids detach from the glycerol and combine with the alcohol molecules, creating fatty acid methyl esters, leaving only behind biodiesel and glycerol as a byproduct. Like bioethanol, it's mostly produced from corn in the United States, and as such remains controversial because both bioethanol and biodiesel potentially could raise the prices if they're used in such mass scales, or if a drought or natural disaster hits that reduces the total amount of grain available. Well, that about covers everything. Thank you for watching. In ancient times, many different species of cacti were domesticated in Mexico. These species were listed on the image seen here, during which time many studies went into trying to figure out how the cultivation of these cacti happened, and as such, three methods were identified. One is gathering fruit from wild populations. The other was a silvicultural management system, in which all but a few species and phenotypes of said species are removed, and the rest is cultivated for food purposes or other uses. The third method is home gardening, where individual plants of columnar cacti were vegetatively or sexually propagated over a long period of time. These processes led to the domestication of these cacti. The differences between the wild and domesticated forms of cacti seem to be almost non-existent, with the only major difference being a higher speed and rate of germination in domesticated populations. For this reason, it's speculated that the domesticated populations have lost much of their drought-tolerant capabilities that they used to have due to being managed for such a long period of time and mass-propagated from singular desirable phenotypes. This paper by Susanna Gulian in the American Journal of Botany was designed to see if this is the case, and here's the results. In the species Stenoceras pruniferus, it was found that the wild populations could withstand greater sunlight exposure than the cultivated ones. The cultivated populations could survive greater levels of high humidity, as well as producing a greater number of viable seedlings in comparison to the wild populations. The wild populations could survive lower humidity than the cultivated form. Further research indicates that seedlings from domesticated populations are 392.9 times more likely to die when exposed to total sunlight and high humidity at the same time, in comparison to wild populations. In both low humidity and 80% shading, seedlings have a 152.74 times greater risk of dying than seedlings of wild populations. In Stenoceras stellatus, it was found that high humidity decreased the lifespan of these cacti across the board in both cultivated and domesticated populations. Same thing goes for complete exposure to sunlight, indicating that the biological limitations are conserved from both wild to domesticated populations. In the species Pelasgia chitpapi, there is no significant differences in survival rates under the different circumstances of high humidity, low humidity, heavy shade, and no shade. In yet another species, Escontria geotila, no differences between wild and domesticated populations were found. During the study, other phenomena were also found. It was found that seeds in domesticated populations were generally larger and heavier than the seeds in the wild population. This may be because columnar cactus species are often used for their fruit. This makes sense since, in cacti, the ones with the largest fruit also have the largest seeds and the largest number of seeds. So by selecting for the largest fruit in cacti specimens, you're indirectly selecting for the largest seeds. The higher rate of germination seems to correspond with larger embryo size, since in cacti, the seeds lack a endosperm, which typically feeds and nurtures the growing embryo until it's able to survive on its own. In the study of the absolute growth rate, there is a positive correlation between seed size and seedling size, with the bigger the seed, the bigger the seedling, with the exception of S. prunosus, which had the heaviest seeds but did not produce the tallest seedlings, or have the quickest growth rate. The relative growth rate did not differ between cultivated and wild forms of the species, but it did differ between different species used, such as S. stellatus and S. prunosus had the slowest growth rate amongst the species selected. In terms of seedling survival, it was concluded that humidity and the amount of solar radiation tend to be the biggest factors in seedling survival in cacti. In the wild forms, perennial shrubs and trees that shade the developing cacti seedlings significantly increase seedling survival in comparison with the open areas. P. chichipe and the geotila seem to be more adapted to drier climates. As such, higher humidity seems to have a greater effect on their health, while the Stenoceras species tend to be more resistant to high humidities, growing in more lakey areas. And in the cases of S. prunosus, cultivated seedlings seem to be more susceptible to high levels of solar radiation than wild forms. For Stenoceras stellatus, there seems to be no difference. This is likely due to the fact that there's a lot of phenotypic plasticity involved in this cactus, meaning that the species can alter its own biology to suit its environment to a very high degree. Well, that about covers everything. Thank you for watching. Hello and welcome to Odds and Ends number 13. In this episode we'll cover the domestication of horse gram, yam bean, or ahippa, and persimmon. You'll start with horse gram. horse gram belongs to the species Macrothyma uniform. Horse gram was the chief crop in India, historically speaking, for most of the history of the Southeast Asian area, with every country within the Southeast Asia region using this as a primary crop for protein sources. However, with the passage of time, horse gram has lost favor and has been reduced to a food of the poor, and there's, up to this point, been very little research onto this topic. Using a paper by Dr. Dorian Q. Fuller and Charlie Murphy, the domestication of horse gram in India will be the topic. Utilizing the ancient, pre-modern Indian and Southeast Asian languages, such as Indo-European, Austroasiatic, Munda, and Dravidian, they found a linguistic history for horse gram. All 4 languages listed have a name for horse gram in their language. In the Dravidian languages, their word for horse gram is kol or kolat, similar to the Indo-European languages such as Sanskrit, which call it kolata, and the Munda/Austroasiatic language calls it koltu, indicating that these language groups are derived from one singular language, and that group of people that spoke that language knew of horse gram and possibly domesticated it. The languages listed, based on linguistics and paleolinguistics, seem to be no older than 1200 BC, and since similar characters were found in the Neolithic pottery, which date back to 3000 BC, the domestication process of horse gram likely started between 3000 BC and 1200 BC. And based on current archaeological evidence, this likely starts sometime in the area we now know as Gujarat, due to the great similarity between many languages of today and the past being spoken of within the archaeological evidence within that region, based on pottery characters. This prototypical culture is now referred to as a Proto-Dravidian people, and is likely this group domesticated horse gram. Biological and archaeological evidence also seems to suggest that, yes indeed, horse gram was amongst the first crops domesticated directly in India. Both wild horse gram and wild mung bean can also be found within the regions where both species are hypothesized to be domesticated, further indicating their domestication was in that region. There also seems to be 2 distinct groups of horse gram, given that, like with mung bean, there are distinct populations of wild horse gram found in the Himalayas and the savanna, and based on recently found archaeological evidence, different domestication trajectories were found in different locations. For instance, in northwestern India, the domestication process began during the Harappan, which began in the year 2600 BC approximately, while another population seems to be domesticated between the year 2000 BC and 1001 BC, indicating 2 separate domestication events for horse gram, again very similar to mung bean. Now on to the domestication of yam bean. Yam bean, also known as Pachycerus erosus, as well as 2 other species such as P. ahipa and P. tuberosus, and all 3 species can crossbreed with each other to produce fertile hybrids. These bean crops, unlike most other bean crops, are not harvested for their pods. Instead, despite being a legume, only the roots are harvested, which are very yam-like in look, and the species has been historic food substances for both Mexicans in ancient times and Southeast Asian people. And to a certain extent, people in Mexico still produce a fair amount of it, with a small area of farmland producing 160 tons per hectare. P. tuberosus and P. erosus are the most often consumed species of yam bean. The tuberosus plants are generally larger than the other 2 species. In an early study of this crop species, using the chloroplast DNA, it is likely that all 3 species derive quickly from one common ancestor due to varying climates, domestication, and human selection. Tuberosus species is likely the ancestral species to the erosus species, although this conclusion was not fully conclusive. Similarly, the ahipa yam bean lacks much of the genetic diversity found in the other 2 species. As of right now, genetically speaking, the landraces and cultivars still have unclear origin, and there are no clear subgroups related to this geographic origin for the different land races. For instance, the Central American and Mexican strains were cultivated by the Aztecs and Mayans, but based on current genetic evidence, they came from different locations within their wild relatives, which have great genetic dissimilarities between them. But based on current evidence, the domestication of the tuberosa yam bean happened in the Peruvian Andes, and has been found all throughout South America, including Paraguay and Bolivia. And although most Pachyhyrus species are very diverse genetically, all the tuberosas yam beans were very genetically similar to each other, more so than any other species within that genus. There's also great genetic diversity within Diarosis, the yam bean, likely due to geographic isolation and domestication. And based on genetic analysis, Diarosis yam bean could be divided into 4 broad groups, based entirely on genetic distinction. One is found exclusively in Central America, 2 groups found both in Central America and Mexico, and 1 found only in Mexico. Yam beans are typically primary self-pollinating, but there is some crossbreeding that does happen, between 2 to 4% crossbreeding, depending on how available pollinators are. Now onto persimmon domestication. The common Japanese persimmon, Diosporos kaki, is one of the most ancient trees domesticated in China. The first known mention of this tree was around 450 BC, when the grandson of Confucius, Kongji, wrote his book Li Ji Ne Zi, which I'm definitely mispronouncing the names of, where it's considered a very important crop during the Warring States period between 475 BC and 221 BC. In another book called Lin Fu, written by a guy whose name I can't even pronounce, between 120 and 118 BC, observed that the West Han dynasty also planted quite a few persimmons. It was only enjoyed by the rich few until the South and North Dynasty era, which lasted between 386 AD and 589 AD. The crop became further popularized due to the improvements in grafting and the ability to remove astringency from the fruit, both methods being developed during the Tang and Song dynasties, which lasted between 618 AD and 1279 AD. Many cultivars were being bred. This is likely when the domestication process happened. It was further improved during the Yuan, Ming, and Qing dynasties, as well as the time period between 1271 AD and 1963. Sometime between these 2 time periods, during the intervening periods between then and modern days, the Japanese persimmon was introduced to Japan in the 7th century AD, and the 14th century within Korea. It was later introduced to Spain in the 16th century as an ornamental. The information spread to Europe in the 17th century and became widespread throughout many parts of Europe in the 18th century. And that about covers everything. Thank you for watching. Hello and welcome to the final episode of Odds and Ends. In this episode we'll cover 3 crops that are relatively common but have very little information regarding it. Celery, arugula, and rhubarb. Get ready for a short video. Celery, or Apium graveolens, is a species native to the Mediterranean, southern Europe, parts of Egypt, and parts of Sweden. It's in Sweden where the forms we know as regular celery, known as subspecies dulce, and celeriac/celery root, known as subspecies rapaceum, are found. It was initially planted for medicine and then later grown for food purposes in Italy, France, and England during the 17th century. Celery, likewise, was introduced and domesticated in China 2,000 years ago. In China, the ancient introductions and their derivatives are called Chinese celery or local celery, while the European American breeds are called just regular celery, indicating 2 genetic origins, one being bred in China and one being bred in Sweden. Based on genetic analyses, both common celery and Chinese celery are derived from the dulce subspecies. Now on to the next crop. Arugula is a member of the genus Rusa. and it's the only member of that genus. The species name is Arusa vesicaria. It is native to the Near East and parts of the Mediterranean, and was introduced to the Americas and Asia, specifically India and Pakistan, and comes in 3 subspecies. Its earliest mention was in the Talmud and the Bible in the Book of Kings, specifically 2 Kings 4:39 to 4:30. The seeds were used historically for deworming and improve digestion. The Italian and Roman individuals also used it for improving the libido. As of right now, nothing is known about the domestication process, but based on its mentioning within the Old Testament, it is plausible it was domesticated in the Near East, although that part's my speculation. Now onto the next crop. Now we're talking about rhubarb. Rhubarb is part of the genus Rheum. The wild rhubarb, known as Rheum repuscum, is a diploid, but the current garden rhubarb is a tetraploid. To figure out the origin of this domesticated form, the researcher Bo Libert studied the history of its wild form. It appears that the wild rheum, or wild rhubarb, has been cultivated prior to regular rhubarb. Morphologically, cultivated rhubarb ranges between looking similar to European wild rhubarb and a rhubarb species called Rheum rhabarbarum, indicating the possibility that the common rhubarb is actually a hybrid between these 2 species. The wild European rhubarb has been found throughout the Onstead Mountains of Norway and the Ria Mountains in Bulgaria. It is unknown if it's truly native to Europe or if it's introduced from somewhere else. And that about covers everything. Thank you for watching. Hello and welcome to Odds and Ends Part 15. In this episode we'll cover the domestication of Noni, Feijoa, and Jamun. We'll start with Noni. Noni is part of the genus Morinda, specifically Morinda certifolia. It's general knowledge that noni is a fruit that tastes terrible, but is highly nutritious, and as such has been used historically for starvation food as a last resort, and medicine. The researcher Anura K. Singh has studied, in a paper I'm referencing right now, the genetic diversity of noni and its center of origin and domestication. And here are the results of his study. Based on the predominance of a high level of Morinda species residing in India, with India the second highest center of diversity of species of Morinda, second only to China, it is very probable that noni was domesticated there. The wild form of noni is also found native in the southeastern and western Ghat region in India. It was also found that within the Indian subcontinent, 3 forms of noni were found: the wild form and the domesticated form, as well as all of the intermediaries and genetic approximations of intermediaries, i.e. hybrids between wild and domesticated forms. All the species of Morinda within India, including Morinda citrifolia, are morphologically and genetically very similar, further indicating that this is where noni was domesticated. There are also writings and diagrams dating back to 1500 BC, showing an ancient use of noni far longer than most of the other Asian locations. There's also evidence of trade between India and China under the barter trade, a practice that's still going on with Myanmar. Based on the current information, Southeast Asia seems to be the primary center of origin and domestication for noni. Now on to the domestication of the Feijoa. The Feijoa is in the genus Aca and is of the species Aca silowiana. It is a species native to the South Americas, predominantly in Uruguay and Brazil, as well as parts of Argentina and Paraguay. Known for its sweet acidic flavor that's mostly consumed raw, but several commercial products contain Feijoa. It is commercially planted right now in China and New Zealand, although this crop is only partially domesticated. Dr. Joel Donalazzo sought to identify the genetic diversity of both domesticated and wild populations to see how much genetic variation there is in both groups and how to preserve the wild populations for further breeding purposes. And here are the results. In terms of genetic variation within populations, it was found that there was higher inbreeding between individuals within the managed populations versus the wild population. Inbreeding in this sense meaning crossbreeding between related trees. Also within the wild populations, there seems to be at least 2 distinct genetic groups within the wild populations, in comparison with 1 in the domesticated population. In terms of between populations, there was found to be a great amount of crossbreeding between the managed populations, which are divided into populations grown by farmers, populations grown in backyards, plants generated from mass selection, which have all continuously crossbred with each other. There also appears to be some crossbreeding between the wild and domesticated populations as well. The Chilean domesticated population, grown by farmers primarily, is the most differentiated population of Ajoa in terms of genetic distinctiveness from the original wild population. Within current genetic analysis, it seems that the Chilean domesticated form is derived from Uruguayan specimens of Ajoa. Now on to its breeding history. After the first European who discovered it, Mr. de Watt from Switzerland, collected and planted it in one of his greenhouses in 1887. In 1890, the at that time renowned horticulturist, Dr. Edward Andre, bought a specimen from La Plata, Brazil, and grew it in the Riviera in his garden. After it flowered and fruited, he wrote a description of the entire plant and recommended that everybody grew it. Later on, a nurseryman in Lyons named Benson Freres obtained cuttings and layer seeds from Dr. Andre and Montevideo, respectively, and raised thousands of plants. Later on, seedlings from Dr. Andre's plants were obtained by Dr. Francisci in Santa Barbara, California. It is from these populations that the domesticated form of was bred. Now on to Jamun. The Jamun is a species of Syzygium called Syzygium cumani. It is native to the Indo-Malaysian region, where it's distributed pretty much everywhere within that region. It's a fruit common amongst these groups of people. As such, the researcher Dr. Safir Ud-Din of the University of Agriculture in Faisalabad, Pakistan, I hope I'm pronouncing that correctly, to find out genetic diversity, and here are the results. Based on the 50 strains from 10 major Jamun growing districts in Punjab, it was found that indeed it was first domesticated in Pakistan, with the area now known as Pakistan being the location of the greatest level of genetic diversity. It was found that the strains labeled SFM08, TUC01, SFG07, KKF02, MWF01 are the most predominant strains that have the best features for future breeding projects. The strains KDC03, TUC02, and TUC03 seem to be strains that are bred for small seeds. It was also found that genetic diversity within the domesticated populations is very small, consisting of only 3 distinct genetic populations. As such, it's estimated that future crossbreeding attempts would be between different domesticated groups to improve heterosis, or between the wild forms and domesticated forms. And that about covers everything. Thank you for watching. Hello and welcome to the last episode of Odds and Ends. In this episode, we will cover the domestication of 5 different tropical crops: Tamarillo, ice cream bean, guarana, Brazil nut, and cupuaçu. You'll start with Tamarillo or tree tomato. The tree tomato goes by the scientific name Solanum bacatum. It's a small tree that's cultivated for its edible, slightly acidic, tomato-like fruit. It's popular as a niche crop in its native home of the Andean region of South America, as well as New Zealand and the Mediterranean. In North America, it's only considered a minor fruit oddity. In the paper I'm citing, Dr. Pablo G. Acosta Quinceada studied the genetic diversity of the species to find out its origins, center of domestication, for possible new breeding strategies in the future. Here are the results of his efforts. Based on genetic information obtained by the researchers, within the domesticated populations, there's high levels of genetic diversity between plants within a population, but very little in terms of genetic differences between land races. It was also found that although most of the wild specimens of tree tomato have been found within Bolivia, high levels of genetic diversity and low genetic differentiation are found within Ecuador, indicating that Bolivia, despite being the center of origin, has less genetic differentiation than what is found in Ecuador, indicating that Ecuador is the center of differentiation between populations for tree tomato. This little variation between geographic origins within the Andean land races is plausibly due to the continuous exchange of seeds between different cultures, which would allow for continuous crossbreeding between landrace populations, preventing genetic differentiation from happening. Now on to ice cream bean. Now on to the ice cream bean. The ice cream bean is a series of different species belonging to the Inga genus. The most common species used is the species Inga edulis, which I'll call IE for short. This is a diploid tropical tree legume that's pollinated by birds and flying insects and bats, and it produces very large beans, specifically bean pods that have their seeds coated in a sweet gel, which tastes apparently like ice cream. The managed population seem to be domesticated, as the Amazonian species, in its domesticated form, has far larger bean pods than does the wild form. Unlike most other species of domesticated crops, the domesticated form of IE still has 80% of the genetic diversity of the wild population, higher than the expected and typical 65%. This is likely because of the high tree density within plantations within the Amazon, and a close proximity to the wild progenitors, which allow for greater genetic diversity within these populations. Now on to guarana. Guarana belongs to the genus Paullinia, and to the species Paullinia cupana. This crop was domesticated in the region between the Lower Tapatos and Lower Madeiras rivers within the central Brazilian Amazonia region. The first European note of guarana was in 1669, where it was observed that the Satri Maui people were the original cultivators of this vine. There is a myth that surrounds this fruit. There was once a woman whose name I cannot pronounce because it's very long. It was basically about a family who guarded their sister jealously because they wanted to keep her knowledge of plants all to themselves. She was also the owner of a magic Brazil nut tree. One day a small snake seduced her and impregnated her, resulting in a snake-human hybrid of some sort. The Tiktov brothers managed to concoct a scheme to kill the kid, which ended with the boy being decapitated. She took out the left eye of her decapitated son and planted it in the ground, producing the false guarana, and then took out the right eye and planted in the ground to produce the true guarana. This myth may ironically have some basis in fact, though probably unintentionally. Based on a study of the genetics of the guarana, it seems to indicate that the guarana is actually a hybrid between two different genuses of the same family, because most false guaranas, or different Paullinia species, have a chromosome number of 24, but the cultivated guarana has 210 chromosomes. The number of chromosomes and the morphology of these chromosomes suggest a combination of a tetraploid and a hexaploid species formed through hybridization. This sort of mirrors the myth based on the work of the paper I'm citing, given that this species is morphologically dissimilar to the other species of Paullinia, or false guarana, and was likely formed through hybridization between two different genera. This mirrors the snake-human hybridization, and the plantain of both the false and the true guarana, although in this case I think that the researcher in question may be reaching a little bit, but still makes for a fun video. As for when the domestication occurred, it is known that 4,000 years ago, within the Andes region, food production systems became more important than foraging. The original planters of this crop arrived in their current location likely 2,000 years ago. This indicates that the maximum time period for when it was domesticated was 2,000 years ago. With current genetic evidence, it may have been even more recent, however, due to very limited genetic differentiation, estimated about 600 years ago, so the time frame of domestication is between 2,000 years ago and 600 years ago. Now on to Brazil nut.